thanks again to the organizer, Luciano and Guyenne, for inviting me to speak about my group's effort around quantifying biological shape across imaging modalities. And thanks to all of you for being here to listen to this presentation. So my group at the European Bioinformatics Institute in Cambridge in the UK is interested in the problem of quantifying morphology or shape from biological images, from microscopy images. And we are interested in this problem not in just one type of microscopy image, but really in every single possible type of microscopy image. And the reason for that, and I'm sure I don't need to convince you of this, is that shape or morphology matters at pretty much all scales of biological systems, whether you look at what's happening inside a cell, at the cell level, at the tissue level, at the whole organism level, actually maybe even to higher scale, like groups of organisms. We won't go that far, but any sort of biological processes has some sort of morphology-related aspect to it. And what you see on this slide are examples of different projects that I really use here to illustrate these points. So these different projects, I won't tell you so much about what they are about, but in a nutshell, they focus on investigating different types of biological questions. So whether it relates to function, motility, development, structure, they are happening at, we are to explore them looking at systems at different scales, and we use different types of microscopy modalities. So, this looks like a lot of different things, but they all have this common denominator that down the line, what matters is shape. So from this, what we are really asking ourselves is there a way to approach this problem and look at this problem in a general manner, such that we can somehow abstract ourselves from the fact that these are all different kinds of data and really go down to the essence of the problem. How do we quantify visual information that relates to morphology? So that's the sort of things that we are exploring. And to kind of see how we are doing that, let's maybe go back to looking at what the process of quantifying morphology for an image looks like in general. And classically, this is really a two-step process. So the first thing we need to do once we have a microscopy image is to figure out which part of the image contains the object, which morphology we want to characterize. This is obviously segmentation, so that's a very classical problem in bioimaging in particular, but in computer vision in general as well. And as I'm sure all of you know, there has been massive development in recent years, thanks to many different kinds of technology, but machine learning in particular. And I would not say that this is a solved problem in general, but we have very good tools to approach this in each and every individual instance where we encounter a segmentation task. So you may recognize some of the tools and techniques that are listed on this slide, but essentially what comes out of the segmentation process is a description of the object that we want to characterize as a collection of pixel coordinates in an image grid, right, in the grid that it came from. And then from this, what we can do is to actually describe the morphology by building a descriptor that will be a collection of numbers. And these numbers can range all the way from like very simple handcrafted metrics, such as, I don't know, the size of the object in terms of its area, perimeter, these sort of things, to more complicated and maybe less interpretable ones that we obtain, for instance, with machine learning. So that's what the general process looks like. Now, there is actually something that we have to keep in mind, is that the nature and the value of the numbers we extract from this region of interest to characterize its morphology actually not only characterize the biological object, but also hold some information about the grid that we extracted them from. So typically things like the number of pixels that we had in the first place, or like their size, so the resolution of the image, are going to influence the value, like the precision of the metrics we can get, but also like the sort of metrics we can compute. And this may sound like a triviality, but actually it is not, because it means that if we were to image the same object under different microscope, for instance, then what we would get effectively is different descriptors with different numbers that would be hard to reconcile, because the sort of information they hold is a mixture between the biological information that we see at different scales and something that is completely unrelated to the biology and that relates to the acquisition system that we've used. So somehow it feels that like this process doesn't really help if what we want to do is to be able to aggregate information that we see and that we obtain from different types of visual observation, for instance, obtained with different microscopy technologies. So acknowledging this, what my group is essentially trying to do is to propose an approach that is slightly different, and in which, instead of relying on pixel sets to describe morphology, we rely on a different kind of objects that are called geometrical models. These guys have, I mean, you'll hear all about them in this presentation, but essentially they have two advantages. The first one is that they are constructions that are continuously defined. So there is no notion of grid. It becomes completely possible to compare things across different types of images, to different images with different properties. And by construction, these objects explicitly describe the geometry of what we had in the initial image. So essentially, it makes our life easier for morphology quantification, because now quantifying morphology boils down to characterizing the nature of the geometrical model we extracted. So what are these geometrical models concretely? Well, they are, for instance, curves and surfaces built by interpolation techniques. That can be spline approximation, that can be subdivision schemes, that can be meshes. I'm sure that you actually know many of them, and you probably have actually already interacted with geometrical models, because these are the objects that are at the core of computer graphics and vector graphics in particular. So if you've ever done vector graphics drawing, for instance, in Illustrator, this is actually the sort of representation that you were manipulating. Sorry. And this is also, so this process of using geometrical models called geometrical modeling is something that is actually used quite extensively in medical imaging for shape quantification. So, we have very good reasons to believe this is the right place to go. However, obviously, the sort of challenges that we encounter when working with microscopy data are not really the same as with MRI data. So there are things that need to be adapted and repurposed for the context of microscopy image analysis. So the beauty of geometrical modeling is that it really gives us kind of the necessary level of abstraction to consider this problem of quantifying morphology in an extremely general manner. And what I mean by this is that we can actually go back to the images I had on my first slide and now see that as variation on the same theme, which is I have different geometrical models and I want to quantify how different they are. And that can be either populations of different instances, that can be either one instance that deforms over time, that can be instances at different scales, that can be instances with different amount of resolution information. So this kind of really gives us a framework that says, okay, now we can consider the problem of quantifying morphology from microscopy images as one big problem, regardless of the actual image data we started from. So that's where we are heading to, but obviously this is a big task. So we are doing that step by step. And what I want to tell you about in this presentation are essentially two sort of tools that we are developing that to reach this goal. The first one fall in the category of pure geometrical modeling. So this is essentially, how do we get geometrical models out of microscopy data? And I want to give you a flavor of what are the challenges, what are kind of the things that are open questions, what are the things for which we have solutions already. And then the second part is, once we have geometrical models extracted from images, how do we characterize them? How do we use this representation to gain insights on shape and morphology? So let's get started with this first bit, and that really kind of would answer the question, how do you go from image to geometrical model? So if I give you this image, how do I get a geometrical model of this cell? And maybe the first thing we can do to get this started is to kind of really get a better feel of what are the elements we need to build a geometrical model. And to understand that properly, and that may be because I spent a lot of time with people from the signal processing world, let's first go back to a 1D problem. So think of how we would build the equivalent of a geometrical model, but on an object that is a one-dimensional function. And then essentially what will happen in 2D, 3D, 4D is going to be the same spirit. So let's assume that what we are looking at is an object that we know is continuous, so a continuous function, but we only have access to finite set of discrete samples. So that would be the points you see on this slide. What we want to do is to effectively continuously approximate the underlying function from these samples. And I mean, I probably won't teach you a lot through that. You can do that very easily by interpolation, by saying, okay, I have these samples. I know some things about the very nature of this function. for instance, I don't know the fact that it is a polynomial of a given order, so I can pick the right interpolation scheme that is good for me and reconstruct an approximation of the continuous function that was there in the background. So that would be simplest case. Okay, linear interpolation, you just draw a straight line between these points, right? This sort of construction is effectively expressed as a model like this. So this would be something that is a weighted sum of some parameters that are functions of the sample points you started from, and the collection of basis functions that, in the simplest case I mentioned here, would just be the linear interpolator in each and every interval. Now, this simple model actually is very rich because we can do much more with that. We can pick different functions for different intervals, or we could do something that is just like still simple, but the next level of simplicity after this one, to just say, oh, actually, maybe, you know, I'm not happy with this approximation. I would like a smoother function. So the only thing I'm going to change is just the interpolation that I use. And maybe now I use cubic spline interpolation and I get a smoother approximation of my function. So that's the spirit. And again, probably you know already this, but then it would make it easier for you to understand what we are doing in images, because the problem that we are looking at with microscopy image is very similar. Effectively, what we have is a discrete set of sample points for the contour or the surface of an object, because I think we will all agree that no cell ever was made out of pixels. So what we see in terms of the pixel content is just a sampling of the actual cell contour or surface. And all we are asking is, you know, how do we use these samples to continuously approximate the shape or the morphology of the object that we are observing? So concretely, there are two things we need to wonder. The first one is, which sort of basis functions do we use? And the second one, in what are the parameters of our model? So these two functions, questions are actually kind of linked to one another. And I just want to give you a feel of this with this little doodle here. So let's assume that is the cell I'm looking at. So that is the set of pixels that I start from. I want to build a model of this object. Well, actually, these two models are very valid in terms of, you know, how much they approximate our sample points. The top one is like, has a whole lot of parameters and somehow linearly interpolates between them. The bottom one has less parameters and somehow a smoother kernel. You know, if you ask me personally, I prefer the second one because it has a flavor of like, you know, what is the minimal amount of parameters you need to express this to capture, sorry, the geometry of this object. But maybe in different applications, you know, you may want another one. So there is a lot of freedom around that. So let's first have a look at the question of the basis function. And here I don't want to overwhelm with too much information here, but in a nutshell, what I want to convey is just that this is kind of an open field of research, as in the question we are asking here really is what sort of geometrical properties do you want the object that you reconstruct to have? And again, if we are looking at, you know, synthetic shapes or buildings or man-made objects, then we can actually make a lot of assumptions on, okay, whether things are polygonal or whether they are circular or whether, I don't know, they are a third-order polynomial, because yeah, they are man-made and they are very constrained. Now, in the context of biological data, you know, can we tell that a shape is a piecewise polynomial, a cell is a piecewise polynomial object or a piecewise linear object? Well, I mean, yeah, that's this is difficult. So let's say that here, yeah, there is a little blur between what theory tells you and what practice tells you. But again, kind of in terms of what's available out there, you know, if you were to segment cells that are in an epithelium, for instance, and that are kind of highly constrained in polygonal shapes, then maybe you may want to use a linear interpolator. If you know that the cells are somehow in a softer tissue and can be smoother, maybe you want to go for the cubic spline interpolator, which is everyone's favorite interpolator generally. If the cells are in liquid, then we have some knowledge of physics that surface tension is minimized when we have things that are spherical. So maybe you may want to go for something that is more an exponential interpolator. This kind of really depends on the application. Again, the beauty of the framework is that you can actually do much more than just these basic types of interpolation that are used also for 1D series. And you can kind of try and think, okay, what would be good construct for specific biological problems? And again, I won't go too much into the details here in this particular talk, but this is sort of what I was working on in my previous life before joining EMBL-EBI. We can actually think of which sort of interpolators do we construct if we want different kind of properties. For instance, an object that has different types of smoothness depending on where it is on the object, or an object that becomes more complex over time, so for which we can add parameters as time goes by. And obviously there is also the question of, you know, how many dimensions do you want? So are you constructing 2D object, 3D object, 4D object, or even more? So let's say this is, I don't have a good easy solution to give to anybody right now on the question of which basis function to use. I'm hoping to come back and tell you all about how we've solved that with AI in a few years. But for the time being, to actually figure out which basis you need to use, you kind of need to either, you know, think a bit about it, or get in touch with people who know about that. But there is a simpler problem that is saying, okay, if we have fixed the basis function, so if we have said, for instance, okay, it's smooth, whatever, it's going to be cubic B-spline, can we figure out what are the right parameters for this model? And can we do that without any prior expertise on approximation theory, just by learning from the data? And this simpler problem is actually very solvable. And my student Soham has told everything about it in a previous instance of this microscopy symposium, so I encourage you to have a look at his talk. Essentially, we have approached that with a neural network by adapting a popular segmentation method called StarDist. And this spline dist technique that we are proposing pretty much does the following. So if you give it one image with many objects and tell it, okay, I want linear models, for instance, it's going to spit out the parameters that are needed to approximate each and every of the objects based on this interpolator. And then you can change your mind and say, well, actually, you know, maybe I want something smoother, and it's going to then give you the right parameters for this other type of model. So this is kind of the first step towards making this whole geometrical modeling approach more user-friendly and more accessible at a large scale. This has so this spline dist method that actually does that has been used now by several people. So oh yeah, sorry, yeah, I have something that went wrong in this slide. I encourage you to have a look at the paper, have a look at some talk, and have a look at our code if you want to use that yourself. And again, because this was previously explained quite a lot in another talk, I won't go too much into details, and we're going to now assume that we have a way to get these geometrical models out of images. So the next question is, what do we do with them? You know, okay, now I have this nice cubic or linear spline interpolated contours. Yeah, what can I do with this? And the kind of use case that I want to use here to illustrate the sort of things we can do is these little C. elegans worms that are swimming around. So we see that they are swimming in different ways. I mean, I don't know, I kind of sense that there is something different going on. But you know, can we quantify that, and can we use geometrical models for this? So if I try to frame the problem a little bit differently, what we want to do here is say, okay, each of the video becomes a collection of geometrical models that represent the position, let's say the configuration that the worm has at this given time point. We have one collection for each video. What we want to do is first investigate the sort of elements that we have in each of the collections. So you know, what are kind of the main modes, the main shapes, the variants, these sort of things. And then can we compare the two collections with one another? So can we say something about how these collections differ? And I just want to say also that all the things I'm going to present in this section is joint work with my colleague Julien Fajot and our former student Anna Song, who actually really did most of the work that you're going to see here. So to solve this problem, we got interested in a technique called sparse dictionary learning that has actually nothing to do with shape analysis in the first place. This is a general data analysis technique. And it essentially goes like that. So we have as input a data set of k elements that are vectors in RD. And what we are trying to retrieve is a dictionary of J elements called atoms that have the same dimension as the input data, along with a collection of scalar weights, these alphas here, that when linearly combined with the dictionary element are able to reconstruct the elements that we had in our collection. So we are minimizing the reconstruction error, as you see here. And if J, the number of elements in the dictionary, is small, then this is a dimension reduction technique, right? This is effectively searching for a way to encode the data with the smallest number of elements. So in a sense, this is something that basically allows you to retrieve what are the main patterns that are present. And in this sense, it may sound very familiar and very similar to principal component analysis, and it has indeed some links to it. There is one big difference, though. It's this sparse in the name and this condition that you see here that tells you that at most N0, which is a fixed parameter, elements can be used in any of the reconstruction. And typically N0 is smaller than J. So we're not only saying, OK, we want a small set of elements that can explain the whole collection of input data. We also say that to explain the input data, we want to use not all of these elements at the same time. And what this enables essentially is to deal with populations of data that are not normally distributed. So things that are multimodal and that may have different modes of variation. So as I said earlier, sparse dictionary learning per se is not linked to shape analysis. This is just a general data analysis technique. And for this reason, it has attracted quite a lot of attention and very good work has been done around that. So it's not a simple optimization problem to solve, but there are very good ways to solve it. The probably most popular algorithm is attributed to Meral and dates from 2012. So then the next question for us is, can we use that directly to figure out what's happening in our little collection of geometrical models? And to answer this, we have to ask ourselves a first question, which is, do we really want to consider the geometrical models themselves as input data? And I hope I'm going to manage to convince you that no, that's not what we want. Because effectively, what we want to characterize is the shape of these geometrical models. And now I'm not using the word shape as we would use in classical English vocabulary. I'm using the word shape as in the definition of KENDALL shape, so in a very specific way. So shape is whatever remains once we have removed simulated transformations. So that would be position, scaling, and orientation from a geometrical object. And just to give you a sense of the fact that this really is the right concept, look at these three leaves. I think we all agree they have the same shape. That's exactly what this notion captures. So these three leaves are different geometrical models because they are placed at different locations. They are rotated in different ways, but actually they are all the same in terms of the morphology information that they contain. So that's literally what we are saying here. So what we want to do effectively is to do sparse dictionary learning, but on input data that are KENDALL shapes. Is it straightforward? Not necessarily, for two reasons. The first one is that the space of KENDALL shapes is something that is very well characterized mathematically, but that is classically defined for input data that are discrete point clouds. So that's actually not quite pixel sets. I'll come back to that in a moment. That's landmark sets, but essentially it's not geometrical models. So it's not straightforward to characterize what the space of KENDALL shapes from geometrical models would look like. That's the first thing. The second thing that is more technical, but also more challenging, is that the space generated by KENDALL shapes is a nonlinear space. It's a Riemannian manifold. So what this means concretely is that we just can't reuse the linear formulation for dictionary learning that I showed a few slides ago, because we have to give a meaning to things such as linear combinations on a nonlinear space. So for this second aspect, the nonlinearity, people have actually looked into that. And the sort of solutions that exist fall into two categories. The first one is to rely on a kernel trick that essentially boils down to mapping the manifold onto a Hilbert space, generally a reproducing kernel Hilbert space. So this works well, but is very heavy mathematically. The other more simple solution is to locally linearize the manifold by projecting on tangent spaces. If you've ever done shape PCA, this is generally what you do. Now, the problem with that is that we are actually distorting the shape space around the place where we project. And while this may be okay for like shape PCA, it is problematic in the context of dictionary learning, because that means we would get atoms, so dictionary elements that don't look anything like the shapes we started from. And that kind of defeats the purpose of, revealing patterns in the data if we can't really backtrack what we see to the original data. So what we've done in this work essentially is to first propose Kendall shape space construction that allows us to consider both landmark type of data, so the classical type of data used for shape analysis, and the geometrical models, and I'll explain that in a second. And this actually allows us to propose simple and mathematically light formulation for sparse dictionary learning on these sort of Kendall shape structures. So to get started, maybe the first thing we need to realize is that, the first thing we need to do is to just characterize, well, what are the type of data that Kendall shape space expects normally. And I was telling you before, these are discrete point clouds. And this is actually this type of discrete point cloud. So if the object we're looking at is a hand, that is what we call landmark sets. So this is effectively a sparse set of points that mark like striking features on the object, and that can be put in correspondence between different objects of this category. So we could be able to landmark a hand, like any sort of hand with this set of points. So this sort of input is just like a collection of 2D points and like, yeah, let's say n of them here in my annotation. Now, in our case, we are using geometrical models, so they are continuous objects. However, as I told you earlier, they are built from a finite set of parameters. So they are built with this sort of expression that is a weighted sum of some parameters and basis functions. And actually, there is something really remarkable to see here. It is that these geometrical model constructions, they are actually they are continuous objects, but they are kind of an intermediate between the discrete and the continuous world. Because if you know which basis function you're using, so let's assume this guy is fixed. This is anyway what we were doing before to extract these geometrical models from images. Well, if that's the case, these two objects actually, in a sense, have the same number of points to define them, right? We need a finite set of parameters here. Actually, these guys they are not exactly the parameters we use, but they are close enough. So we do need really the same set of points, but thanks to the interpolator, we can construct the continuous object out of it. So in a sense, it's like with the same amount of information plus one bit that tells us what's the basis, we get the full outline for free. So that's the first thing we notice. Now, to build the Kendall shape space, let's remark one additional thing. That is, again, something that I guess I won't teach you. It is that the real plane in 2D can be, the 2D plane can be associated to the complex plane. So I can see a 2D vector as a complex number, right? Where the real part is the x coordinate, the imaginary part is the y coordinate. Let's do that, because this is going to make our life immensely easier to actually derive this whole construction. So now our inputs are something that we call the configuration. This is a vector of complex numbers, right? And the two examples are the two constructions that you saw in my slide before with the hand fall in this category. And we associate to this object a complex inner product that depends on a matrix psi. And this guy, and this guy is essentially going to tell us whether we are in the discrete or the continuous case. So if we are in the landmark case, this guy is going to be identity. If we are in the continuous case, this guy is going to be some function of the basis. Why did I switch to complex numbers? Well, because if you remember what we wanted to do in the first place, we want to consider the Kendall shape of objects. So we want to consider what remains once we have removed position, translation, scaling, and orientation. And this becomes actually quite easy to formulate now that we are in the world of complex numbers. So I want to convince you now that what you see in this box actually characterizes all possible similitude transformation of this input object. Why? Because A is a complex number that is different than 0. So multiplying my input by A effectively captures all the possible effect of scaling and rotation, scaling thanks to the modulus of the complex number, rotation thanks to its phase. And the only thing that remains is the effect of translation. So for that, I just have to define what I call the unit configuration, which is the input vector that allows me to shift an input data by 1 unit. And then with this, I capture all of these transformations. But I can also now very easily define what is a transformation that has been centered, because this is any input data that has an inner product with the unit configuration that vanishes. And I can also easily define what are the input data that have been normalized, because these are the guys which inner product with themselves is 1. So from that, we can build what we call the pre-shape sphere, which is the space that contains all the configurations that have been centered and normalized. So yeah, nothing too crazy here. But then it actually also becomes kind of straightforward to build the final shape space, which is this space where we removed the effect of rotation. But again, because we are in the world of complex number, what are rotations? This is just complex numbers of modulus 1. So we just have to quotient out complex numbers of modulus 1, and we are done. Now, there is one thing to notice, and I won't go too much into details, but effectively the structure of this KENDALL shape space is very similar from the structure of the pre-shape sphere. They have shared minimizers, and what this is going to help in the sort of formulations that we get in a few slides. So with that in mind, we can propose a sparse dictionary learning formulation for KENDALL shapes. Now, there are a few things that changed from the linear formulation you saw before. The first one is that our input data are pre-shapes, so configurations that have been centered and normalized. This is simple operations. The dictionary elements are pre-shapes themselves. The weights are complex numbers, because now that's the world we're in. And what we are trying to minimize is the reconstruction error between the KENDALL shape of the input data and the KENDALL shape of the reconstruction. And that's what these brackets mean. So that means KENDALL shape. We can use a bunch of different distances on the shape space. And let's just now have a look at what we could use. So there is obviously the geodesic distance, so really the distance on the manifold between these 2 points. And we can notice that this guy is a function of another distance called the Procrustes distance that some of you may know. It's also called optimal alignment distance sometimes. So this is the distance you would get if you were to kind of manually align the shape on top of each other. So that's the one that is very straightforward to think of with the leaves that I had before. If you manually align them on top of each other, you'd get 0. Well, actually, this is a bad example but anyway, that's effectively what this distance captured. That's the intuitive one where you align shapes. And what we can show, and again, I won't go into detail here, but I invite you to have a look at the paper if you're interested. What we can show is that this full Procrustes distance, that is a distance between shapes, so on the KENDALL shape space, has actually a closed form expression in terms of pre-shapes. And just as a reminder, pre-shapes are just centered and normalized input data. So these guys are pretty straightforward. And it is then interesting to notice that if we choose to use this full Procrustes distance, so the kind of optimal alignment distance, the formulation that we have boils down to a linear one. So it kind of means that actually, by considering this distance and the construction that I've just showed you, we go back to a problem that is linear, for which we can use the classical framework to solve this optimization problem. The only thing we have to pay attention to is that now we have complex numbers everywhere. So yeah, there is a little bit of change to do, but not much. And in terms of algorithmic details, I just want to mention also, I forgot to put this on my slides, but we have code available. So have a look at the paper. There is a link to a Git repo there if you're interested. But effectively, what we had to do is to adapt this toolbox called SPAMS that is developed by Meral and colleague to solve the classical dictionary learning problem to the case of complex numbers. And for those who are interested, essentially, this is a dual minimization problem. So we alternate between one step in which we update the dictionary elements. So we fix the weights, find the atoms. This is a gradient descent. So that can be solved with a method of optimal directions. And once this is done, we go through the next step, which is saying, okay, now we fix the dictionary, we update the weights. This is called sparse coding. So that's the actually encoding step. And this is done with or the recursive matching pursuit that has to be adapted here again to complex numbers. And we alternate between these two until we converge. So that was a lot about the technical details. So now let's go back to the worms we started with. So why did we ended up doing Riemannian manifolds while what we wanted is to quantify worm motion? Well, I mean, now it will all fall back together. what we started from is these videos of worms at different swimming, and that are actually aged, that have different age. So these guys are younger, these guys are older. We see that there is somehow a difference in their swimming pattern. What we had done is to extract a collection of geometrical models, and we were wondering, how do we compare these shape collections? Well, now we have a tool that allows us to say, actually, I'm going to now summarize this whole collection with a shape dictionary here of 5 atoms. So the first thing that this gives us is a very immediate way to see what kind of features in terms of morphological configurations are present in each of these collections. But it's actually more powerful than that, because now it gives us a very, very compact way to characterize everything in this collection, because each and every little worm now becomes a vector of 5 numbers that are the weights we need to use to reconstruct this shape based on the dictionary. And it's actually less than 5 numbers because there is the sparsity constraint. So several of these numbers will be 0. And so this gives us a direct tool to quantify the dynamics of the worms, basically just by saying, okay, I'm going to look at how this weight vector evolves over time. So that's what you see in these matrices, and this is now color-coded. It's obviously symmetric. And each entry of the matrix tells you how similar the weight vector, so effectively the shape of the worm, is at the time given by the row index and the time given by the column index. And what this reveals is that the difference that we kind of felt by looking at the video effectively comes from the fact that these younger worms, they are building their shape by alternating between very different types of combination of dictionary elements, whereas the older worms have this behavior where they kind of transition between bouts of behavior where they reuse the same atoms over and over again. And then what we could do as well is, just to kind of compare the dictionary elements themselves and have a look at how these two dictionaries overlap. But for that, I want to give you another example involving worms, where the context here is a little bit different. So we are not looking at dynamics anymore. I mean, kind of, we're not looking at dynamics of a single individual. So these are not C. elegans worms. By the way, these are planarian flatworms, so it's still worms, but a different kind. And we are looking at the development sequence of these Dorothecephala flatworms. So this is a population. Okay, here I just show 6, but there are actually much more, of worms of 5 days old. This is a population of 7 days old, 9 days old. And here there is no notion of tracking. So this individual, I have no idea whether it is even present in the population at 7 days. It may be there, it may not be, I don't know. So what we are asking now really is the sort of variability that I see in each of these developmental stages, how do they relate to one another? So we can do the same pipeline again, let's geometrically model each and every person here, a worm person, and then let's build shape dictionaries, this time, okay, 4 elements. So what we see first and foremost is kind of something that we sense already from the geometrical models, that what they do over time is grow these little ears here. We see that quite well in the atoms. But what we can do now is to actually use these shape dictionaries to quantify how much the shape information we have at a given time point, at a given age, allows us to understand, or let's say more precisely, reconstruct the shape information we have at another age. So that's what you see in these plots. Essentially, obviously, the age we are using, so the age we are considering is the one we can reconstruct the best, right? So if we use this dictionary and try to reconstruct this collection, obviously it works well, then it works better than each and every other possible collection. But more interestingly, what this shows us, and this is also kind of an intuition that I think we can easily get from the data, but that is here quantified, is that what's happening as these guys age is that their shape become more and more specialized. So effectively, the older they get, the less able the shape collection is in explaining or reconstructing what was there before, just because it has become more and more specialized and complex. Yeah, so with that, I'm getting to the end. These were a bunch of examples around geometrical modelings, around shape analysis, but effectively, if there is one take-home message that I wanted to have here, is that the reason why we are doing all of this is to be able to one day tell ourselves, okay, how do we quantify shape from these different images? Well, this is all going to be the same, or let's not say this is all going to be the same, but we can approach all of these different problems as one big similar problem, that is, how do we quantify variability in geometrical models that are abstract mathematical construction that therefore allow us to take a step back from image format and actually consider all of these data together. So with that, I want to thank you all for your attention. I want to thank my team for their hard work. I also want to mention that we have postdoctoral fellowships coming up. So if you liked what you saw and you would be interested in working in that, please get in touch with me. And I would be happy to take any questions. Thank you.
All right. Thank you. Thank you, Virginie. I really appreciate your presentation. I don't know if there'll be questions, but I do have one question for you. A lot of the work, and excuse me that this is a little bit kind of a tangent from what you normally do, it seems to me. Have you considered, or are you already using this kind of approach to compress data or to encode data? Because it seems like there's really good potential there in the modeling. You know, you're modeling structures, complex structures into simpler versions of them. And it's such a huge problem, as you know. I mean, there's such a huge problem with huge data that often has little content. And you seem to have here a good approach to extract the content and compress it down to something, yeah, to the essential bits.
Yeah, so I mean, this is a great question, and thank you for bringing this up. It's actually so it's actually not that tangential. So this is very much something we are working on. And when I was talking about at the very beginning, when I was talking in the geometrical modeling part about the fact that, you know, there are different kind of models, and some of them are less parameters, and that's kind of the ones we like. Actually, there is a lot of ongoing work that goes into this, what we call minimal models. So effectively, yeah, what is the minimal amount of information you need to encode the geometry that you actually need further for quantification, or really just for storage, right? Because if what you're interested in is just the geometry of, I don't know, some cells in a light sheet image, then why would you store the whole voxel data when you can just store the, I don't know, 100 times 3 parameters that define the object. So no, yeah, this is definitely an area that we are exploring, and it's a very exciting one. Yeah.
Okay, looking forward to seeing more on that. Good. I see. Let's see. Are there other questions for Virginie right now? You can of course always post them here. Oh, there's one. Yeah, go ahead.
Hi, Lucas. This is Shalin.
Hi.
From BioHub. I lead a platform here. Oh, so there's a amazing talk. I really enjoyed the talk, especially the idea of the shape dictionaries. And so I had one question around it, which is that in some cases, specific types of shape heterogeneity matter. For example, when the cell is growing, its size matters. And when the cell is migrating, its orientation matters. It's biologically meaningful. Is there a way you can choose the invariants of the dictionary? That's one question, and I have a follow-up question after that.
So to that, thank you. That's a very good question. So you can choose to some extent. As in, you know, typically within these 3 transformations that I was talking about here, which are the classical ones we want to remove, you want to like add, or I mean, you want to consider them all or a subset of them. This is very well feasible. It becomes more complicated if there are like additional transformation you want to include, like, I don't know, affine transformations, reflections, these sort of things. Then okay, the construction may become more complicated. But yeah, there is a bit of freedom around that. Yeah, absolutely.
Yeah, I look forward to reading. Then the second question I had was, is there a notion of texture dictionaries that is an extension of shape dictionaries?
So I have not, I mean, I have not personally worked on that, but effectively, as I was mentioning, the shape dictionary, the sparse dictionary learning problem is initially formulated for input data that are vectors. And so in this sense, if you wanted to learn a texture dictionary, I think you could use right away the classical formulation, because you could, you know, I don't know, you could describe the texture with local binary pattern or any sort of, texture descriptor that computer vision likes, put them into a vector and then kind of use that in the classical framework. So yeah, that would be amenable to that. I think we've not worked on that specifically, but it could very well be used for this as well. Yeah.
Okay, great. Thank you very much. If there are other questions, we can of course you can of course just post them on the chat, and we will either get them to Virginie or she will answer directly in the chat if she has an opportunity. All right.
Can I ask one question, Luciano?
Yeah, of course.
Is there time for a question or not?
Yeah, go ahead.
Virginie, this was a really great presentation. I was wondering how, so with regards to the sparse dictionary learning approach that you describe, how easy is it to do these shape descriptions in 3D? Sometimes, the math works out and generalizes easy over multiple dimensions, but the computation or the implementation aspect can be particularly hard.
Yeah, that's an excellent question. Thank you for that. So actually, like parts of the math generalizes easily-ish, but not all of it, because like everything that I showed, it heavily relies on this thing that rotations are well expressed with complex numbers, right? And so for 3D, you have the equivalent with quaternions, but somehow, like not all of the analogies work. So actually, even just the theoretical framework in 3D is not trivial, and we are working on it, but it's not like it's not an immediate extension. Now, in terms of computational cost, what I'm actually so again, it's a bit too early to tell, but these. I mean, as I hope I kind of, I went very fast on that, but I kind of conveyed on the algorithmic part. Actually, in the end, the algorithm itself is quite light, right? This is effectively some sort of gradient descent. Okay, maybe the gradient itself is complicated, but we now have like kick-ass stochastic gradient method thanks to AI, so this is not really a problem to solve. And in a sense, I'm really, for what we know now, I'm really expecting that this is going to be the case in 3D as well. So of course the data are bigger, but the optimization problem itself, once it's formulated properly, is not so hard to solve. So I would not expect that the complexity really blows up in that respect. The question is like whether we can get to a simple formulation like that. This I don't know yet.
Great, thank you.